This is the second part of Lecture 3 preparation. We're going to move now. We've talked about some of the experiments that first told us DNA was the genetic material, and then one clue to how the molecule was organized. And then Watson and Crick in the 1950s, actually, I had that wrong. Took data with X-ray diffraction and the work of Chargaff, which we've talked about already, and they determined that the molecule was a helix. And then they figured out sort of more details of the structure. And so things to point out is that A and T pair. So remember, A is a purine and T is a pyrimidine, so you have the 2 ring and 1 ring, so the larger and the smaller one paired. And then G and C pair with the larger ring G and the smaller ring C. And we'll talk about the sizing of that in a minute. These are base pairs. A and T have 2 hydrogen bonds between them that hold them together, and G and C have 3 hydrogen bonds that hold them together. So the G and C binding is a little bit tighter, a little bit stronger than the A versus the T. And then something else to just notice, think of this like a spiral staircase. The steps are the same width all the way along, and it spirals around, and there is a polarity. So the 5' phosphate is sticking out at one end of the molecule, and then at the other end of that molecule is going to be the 3' hydroxyl group, and the molecules line up anti-parallel. Oops. Which means that the polarity is the other way around on the molecule that's bound. And so there's the 5' phosphate at this end, and then the 3' OH at this end. And the fact is that if you tried to line them up with the 2 5' ends on the same end of the molecule and the 3' at the same end, these the way the molecules are arranged, these nucleotides would not be able to properly bind with each other. So you can only get this anti-parallel type of binding. So, and the structure that they came up with fulfilled all the kinds of requirements you would expect of a material that's hereditary material. So you would have to have an ability to store information, and if you look at the sequence of DNA, in different genes and so on, the DNA sequence differs. And so looking at different combinations of A, C, T, and G could give you different forms of information. It had to be able to mutate, so, change, a nucleotide, the other nucleotide changes, and suddenly the information is no longer the same. So that would be the way you would mutate it. And then the ability to be replicated. And so they knew DNA replicated, and they worked some time on figuring out the exact way that occurred. So these things I've already pointed out, especially the anti-parallel nature. And the idea is, if you look at this structure and then you imagine, suppose you open it up and have the 2 strands separated, you could replicate both strands by simply building new DNA with nucleotides that are complementary, that bind properly. And if you did it correctly, then you would end up with 2 strands that were the same. So for example, 3', and I'm just going to put, this is one strand, T, and then A, and then A, and then C, and then on this strand, 5', A, T, T, G. And then when you build the new strand, then your new strand would be built with the complementary nucleotide, which is G, which is T, which is T, which is A, and then this one would be built with the complementary nucleotides T, A, and C. And now you have two new pieces of DNA, each of which has the same sequence, right? It's still the same sequence as the original. So that was the idea. And but even though it made logical sense, there had to be some work that showed that that was the way the replication process actually worked. And that's what I've just said here. The idea is that the fact that you had this double helix structure and you had this specific kind of bonding suggested that this might be the way DNA is replicated. And it turns out that is correct, but they had to demonstrate it. So if you start off by not assuming you're correct, but by just asking yourself, what are all the different ways DNA could replicate? Then there are three different methods. There's conservative, there's semi-conservative, and there's dispersive, and we'll go over them in a second. But the conservative replication would be you'd start off with two pieces of DNA, and then you'd have, after replication, a pair of old strands, and then a pair of, I'll make these ones dashed, new strands. So that you had the old DNA as one of the chromosomes and then a whole new chromosome made, right? You have semi-conservative, which is what we just looked at here, where your old helix opens up, and then each of the new ones that you make has one old and one new piece of DNA. And then you have dispersive, and that is where the new DNA is synthesized in here and there, not confined to a particular strand. So the new DNA might look like that would be one of the strands or one of the pieces of DNA, and the other one would have sort of new in patches and the old in patches and so on. And so, you know, it's not obvious what the mechanisms of these would be, but they had to be properly eliminated from consideration, right? You don't want to make an assumption and then do a ton of work based on that assumption and then find out, oh, okay, you know, we were wrong because we thought we knew what the most logical way was, but actually the cell is doing something very different. Okay, so the way they decided to test this was they grew E. coli in a medium that had an isotope of nitrogen that is what we call heavy nitrogen, nitrogen 15 instead of nitrogen 14. And then they made cells where all the DNA had the heavy nitrogen, essentially. They had to grow it for multiple generations. And then they switched it back to normal medium for a couple of generations. And they took after each of the generations, so that's DNA replication and division of the cells to produce new daughter cells. And they took aliquots at different time periods after one division and after two divisions to separate the DNA molecules. And the reason why they did this part is that you could isolate all the DNA from the cells and then spin it in a centrifuge through cesium chloride, which is very dense. And when you did that, the heavy DNA would all end up in one position, and then the lighter DNA would end up in another position. And you could actually tell what molecules you had generated based on where it ends up in the gradient. And they stained it with stains that showed up. So when you were looking at the tube, this was a lot of spinning. They used to spin for, I think, over a weekend. It took a long time for the molecules to separate out. Yeah, and so they would know based on controls exactly where the light and the heavy DNA should be, and then they could tell what their molecules look like. So you want to think about what would you expect to see for each of these models. If you labeled heavy DNA and then you had new DNA that was made with the lighter media, what would you expect in semi-conservative? What would you expect in dispersive, and so on? What would you expect to see? So these are the predictions. So with semi-conservative, you would expect each molecule that it separated, and the new DNA that was made with the lighter nitrogen isotope. So you would expect that all of the newly made DNA would be intermediate, right? If conservative replication was the method, where again, we don't know how that would happen, but suppose that was, then this is what you would predict. You would predict that one of the new chromosomes that resulted would be the original, the old one, and then the other one would have all the new strands, and so you'd have heavy and light. So you would see something different in the tube when you did this. And then the dispersive, again, it would be sort of, little bits and pieces of new and old kind of at random throughout, and you'd expect intermediate bands. And then after the next generation, we would expect, when you see the results, you'll see why we're only looking at these two. After the second generation, the dispersive model would give additional intermediate bands. And so if we just look at, so this would be sort of where the heavy should be, and this would be, where the light should be. And then after the first generation, you would get this intermediate band, which is sort of right in the middle here. So that's what they would get. But then all of these molecules, they're intermediate. They're not heavy or light, but they have more light. The yellow ones are the light. And so in the next generation, the molecules would all have the same density again, but this time they'd be intermediate between the previous band and the band for DNA that's made of only light. So it would be higher up a little bit. Whereas with the semi-conservative model, so these are the controls. We know where these are, and we expect intermediate. So this looks exactly the same as this one. But with the semi-conservative model, then we still have this old strand and a new strand that's made that's light. So this is the same density as this. So we'd have two molecules that look like this. But then these two would be made with this light strand, and then new DNA would be made with the light, with nitrogen 14, and this light strand a new DNA would be made with a nitrogen 14. And so you'd have two bands in this case, one of which was the same as this, and the other of which is the same as that. Okay, so think through these experiments and make sure you can interpret them. There will be some questions on them during the lecture to make sure that you understand. So we're going to look finally at the mechanics of the replication process. So DNA polymerases are the enzymes that add nucleotides to a growing strand at the 3' end. So they add nucleotides by connecting the 5' phosphate onto the 3' end of a pre-existing molecule. Primers are required, and this is because DNA polymerases are not able to suddenly start building a piece of DNA. They have to they can only add to an already existing molecule. So so the cell has to make little RNA-based primers that get the process started. The replication, of course, happens on both strands, but because of the direction of the DNA replication, always adding to the 5' end. One of the strands is called the leading strand, and the replication occurs more quickly and more efficiently on that strand. The other strand is called the lagging strand, and it has to be made in smaller pieces that then get stuck together, and we'll see why in a moment. And of course, the original strands are separated, and they're used as the template. The template is something you use to copy to make, in this case, new complementary strands. Okay, so here's the idea. The DNA opens, if we're looking at a piece of DNA, it sort of opens. Sorry for the messy, right? But the lines here are the complementary base pairing, and then it opens up. Now, the part we're looking at, the replication fork, we're just only looking at what's going on one side of this, and we'll maybe come back to it. I think I will do a third recording just to make it a little bit, give you a little break in between all these kind of long recordings. But basically, the DNA is opened. We'll talk about that, and then a new DNA is made. So you have to have a little RNA primer. First, let's change color so that we know what we're talking about. So there's a little RNA primer, and then new DNA nucleotides are added to the 3' end of that. On this side, this is the leading strand here, we have an RNA primer too, and then the DNA nucleotides are added onto the 3' end, and that process keeps going. Now what happens as the procedure goes on is this opens up more, the DNA unwinds and opens up more, and when it does, this can just keep going. This can just keep adding and adding to the 3' end. But when this opens up, it requires a new RNA primer and then adding nucleotides to the 3' end of that. And then it opens, even further, another primer, and so on. So the DNA on the lagging strand is replicated slower because it's done in pieces. And then there has to be some modifications and some connecting of those pieces together so that you have one continuous strand across the whole chromosome. Okay, and this is where, I think I'll start a new recording here, because then we can go through all the steps, and I'll try to point out all the most important terms. And I know I said to learn the bold terms, but if I've underlined it and changed color, that's also drawing your attention to it. So I will want you to know, what those are as well. I'll try to go back and bold all them in the notes that I post for you.